During the 1860s, this man, named Gregor Mendel, decided to carry out the first genetic experiments by growing pea plants in the garden of a monastery in Czechoslovakia, where he lived as a monk. Mendel wanted to learn what would happen when pea plants with different, easily distinguishable physical traits, such as pod shape and color, seed shape and color, and length of stem, were bred with one another. Mendel chose to work with pea plants because they grew to maturity rapidly. And although peas normally were self-pollinating, it was a simple matter to cross-pollinate or cross-fertilize them with other plants. To carry out cross-fertilization, Mendel collected pollen that carries the male reproductive cells from the stamens located in the flower of one plant and transferred it to the pistil, the female reproductive organ located in the flower of another plant. As a result, seeds were formed that contained inheritance factors from each parent plant. Mendel's first experiment was to see what would happen if pea plants with short stems were cross-pollinated with other short-stemmed plants. The results of this cross did not surprise him, for in the first generation, called the first filial generation by geneticists, only short offspring were produced. Next, Mendel decided to see what would happen if pollen from tall pea plants was used to pollinate other tall plants. This time, his results were not clear, because although the seeds from some tall plants produced only tall offspring, seeds from other tall plants gave rise to first-generation offspring that were a mixture of both tall and short plants. Mendel could not understand these results, so he performed another experiment. He planted just the seeds from tall plants that had produced only tall offspring. He used these pure-breeding tall plants as one set of parent plants and cross-fertilized them with consistently pure-breeding short plants. The outcome of this experiment was just as surprising as the first, because no short plants at all grew from the seeds resulting from this cross. The shortness trait completely disappeared in the first filial generation of plants. In a further attempt to make some sense of these strange results, Mendel cross-pollinated these tall first-generation plants with one another, and their seeds were grown into second-generation plants. This time, Mendel found that exactly 1/4 of the new second-generation plants were short, and the other 3/4 were tall. And these results did not vary, no matter which of the pea plant's 6 physical traits he was looking at. To explain how cross-pollination affected plant height, Mendel postulated that each plant must contain 2 inheritance factors. Plants with 2 tall inheritance factors will be tall, or with 2 short inheritance factors will be short. Yet if tall and short factors are combined, the new plants will always be tall. In other words, Mendel believed that the tallness factor overpowered the shortness factor in these plants. Therefore, he stated that the tall inheritance factor is dominant and the short inheritance factor is recessive. A short time later, Mendel's inheritance factors were renamed genes, and genes would become the focus of tremendous biological research in the years that followed.